Okay, sorry about that. So we have a couple of different types of neurons that we're going to see. So we have sensory neurons. That's the first type. And we have interneurons, which connect two neurons together, and we have motor neurons. So sensory neurons basically are going to be carrying nerve signals from the sensory receptors in your peripheral system to the central nervous system. So for instance, these will basically collect all the information in your body about what's happening in your environment, warm, cold, things like that. An interneuron, however, will basically only be in the central nervous system. And so basically, oftentimes, what will happen is it'll receive the information from the sensory neurons, and then it'll relay it to a motor neuron, right? So for instance, it'll basically receive it from one neuron, and it'll basically be a relay to another neuron. And then it'll basically pass this on. So it'll kind of collect a lot of different inputs from a lot of different directions, and then it'll basically pass this on to the motor neuron. The motor neuron itself then is going to be carrying impulses away from the central nervous system to an effector, like for instance, a muscle or an organ or something like that. And this will basically be delivering that sort of response into the environmental change to your muscle. For instance, if something is hot and you want to move your hand, then you basically take in that sensory information. Your brain decides to move your hand because you don't like the heat, and then that tells you, then the outgoing message, the motor neuron basically tells your hand to contract the muscle so that you can pull it away. Now, neurons themselves are all composed of a couple of different parts. First of all, you're going to have the cell body. So that's one piece that it is actually attached to. You also have dendrites, which are incoming signals, and axons, which are outgoing. So the cell body basically has the nucleus in it and other organelles, just like we learned about any other cell that we learned about in this semester. The dendrites are short little extensions that basically are attached to the cell body. And so this is basically where signals come into the actual neuron. So this is basically the receiving signal. And then the axon is the portion of the neuron is typically a lot longer than the dendrites, but this is the one that basically will conduct the impulse out to the target cells. For instance, individual axons oftentimes are referred to as nerve fibers, and a collection of them will basically be a nerve. These will be taking instructions out to the rest of the body to basically communicate to the rest of the body what the brain wants it to do, whether it's to contract a signal or something of that nature. So that's kind of what the structure looks like. So you can see here is your cell body. So basically that's where your nucleus is. And then all these guys out here, these are the dendrites that are associated with the nerve. And then the long fiber that leaves the cell body is going to be the axon. So here you can see an axon, which is relaying a message to a cell body. So basically these incoming messages coming into through the dendrites as well, being processed by the interneuron in this case. So here's neuron number 1. And then this is the interneuron, which is neuron number 2. And then what's going to happen is it's going to sum all those messages up, and it's going to send that through its axon, and it's going to connect that to another neuron. So in this case is the motor neuron, which then will basically take this out to the muscle, for instance, if this is a contraction signal. So you can see how you have basically a hookup of multiple different neurons basically relaying information to each other and communicating with each other as well. So the myelin sheath basically is something that covers some axons. Not all axons are covered in the myelin sheath, but basically these in the peripheral nervous system are formed by what's called the Schwann cells, which essentially has a lipidy type of a myelin substance, which is essentially their plasma membranes. And what you do is you wrap this plasma membrane around the axon, just like you wrap a piece of plastic around a wire to insulate it, about 100 times or so. And so what you get is this nice thick wrapping of insulating membrane around the axon, and this insulates the signal, this electrical signal. And so basically, each of these Schwann cells will myelinate a part of an axon. So you leave little gaps in there, and these gaps are what's called the nodes of Ranvier. And these are important because these gaps allow for propagation of the signal. Of your nervous signal will basically go down these myelin sheaths. So here's kind of what it looks like. So you can see you have your axon itself, which is basically propagating the signal, and this thick myelin wrap that insulates that sheath, and then little exposed pieces, which are the nodes of Ranvier, in between these myelin coverings. And so this is kind of what it looks like as electron micrograph of an actual nerve. So you can see this is the axon component here, And then the myelin sheath is this nice insulated region around it. Okay. If you have a long axon, usually you'll have a myelin sheath. This allows for nice long distance propagation. If you have a short axon, oftentimes you don't. So this is how we break our central nervous system and our nervous tissue up into gray and white matter. So gray matter basically is essentially an area where you don't have any myelin associated on your axons. White matter is basically a myelinated area. And so when you have all of these lipids together in one area, so a big cluster of these lipids together in one area, it basically creates a kind of a whitishness associated with that particular area. That's what we call a white matter. So in multiple sclerosis, for instance, this is a scenario where you have a syndrome where you have the breakdown of myelin and the neurons can't transmit information normally, and you get basically dysregulation of the signal of your nerve signal. And so ultimately, the myelin sheath is also important whenever you need to do nerve regeneration if a nerve has been damaged in the peripheral nervous system. And so if your axon's been severed, then the myelin sheath basically remains. It will create a passageway for that new nerve fiber to grow into. So it kind of gives it a little bit of a safe passage. Okay. So now the physiology of the neuron. So basically the nerve impulses are essentially these electrochemical changes that occur that move information throughout the nervous system itself. And essentially it's a voltage. And so the voltage essentially we refer to as the resting membrane potential. Potential is basically a synonymous term for voltage. And so the membrane potential of a neuron at rest is essentially the voltage that we record in millivolts. Roughly, it's about minus 70 millivolts of the membrane that is at rest. And this is because around the membrane, we have more positive charge on the outside of the cell and more negative charge on the inside. This creates a charge separation. That's the polarization piece of it, where you essentially have positive charge on the outside. This is due to ions that are being pumped out there, sodium in particular. And you have a negative charge on the inside because you have a relative deficiency of those ions in there. So this is kind of what it looks like. So your membrane, because of the sodiums that are positively charged, are on the outside. This creates a positive look on the outside relative to the inside. So that creates a negative 70 millivolt potential. So it's a very slight voltage, but it is a working voltage. And the way that you create this is by basically pumping sodium out into the outside of the cell. That creates positive charge buildup, which is why it becomes positive on the outside. On the inside, relatively speaking, it's not as much positive charge there, so it looks kind of a little on the negative side. And so you have all these gated potassium and sodium channels that allows essentially for the passage and regulation of this membrane potential so you don't get too much buildup out there. And so the neurons will maintain this resting membrane potential, and it's basically created by a special pump called the sodium-potassium pump, which will pump your sodium out of the cell and potassium into the cell of the neuron. And so the important thing is this sets up the scenario that we need in order to create what's called an action potential, which is an electrical signal that essentially allows neurons to send messages from one cell to another. So ultimately, a stimulus, which is basically a change that will turn the neuron on, will allow it to basically send the action potential. For instance, a stimulus for pain in the skin would essentially create a neuron activation. And so like you might get from a prick or a sharp pin or something of that nature. Typically, when you're looking at it, you're not just looking for the movement of these ions, which is how you generate an action potential. So it's basically generated by the movement of ions, in particular sodium ion. But there's a threshold effect. So that is to say a minimum voltage that you have to get to in order for your action potential to turn on. This generally is right around minus 55. So you have to go from resting, which is minus 70, to minus 55 in order to get to action potential. And that's kind of like a firing of the nerve. So it's kind of what it looks like. So oftentimes what will happen is the sodium channels will open up. That allows sodium to inrush into the inside of the cell. And that what that does is basically loads positive charge up in the inside of the cell. So it's a charge flip. That's what they're showing here. So this is what's called a depolarization, is when you get that charge flip because you have the inrush of sodium. That changes the voltage. So now you're bringing that down to minus 55. So it's not quite as much potential there. And then once you get to minus 55, then all of a sudden you basically reach that threshold. Your action potential happens, and generally it happens completely. So it doesn't vary in size or amount. It basically happens completely. And so this action potential is an all or nothing sort of thing. You either open up the floodgates or you don't. There's no in between. And so ultimately, increasing the strength of the stimulus doesn't change the size of the action potential. So you can't actually change the size of the action potential by changing the stimulus. It's either all or nothing, right? It's basically you're all in or you're all out. There's no modulation there. And so ultimately, in order to convey a stronger signal, what you do instead of increasing your action potential is you actually create more action potentials in a unit given amount of time. So the concept of depolarization is this idea where the sodium rushes into the cell. That creates a charge flip. In the cell. So all that positive charge now is on the inside. Remember before it was on the outside. And so now all of a sudden the positive charge is on the inside, and that's what we refer to as depolarization. So the charge inside the axon is going from negative to positive. So that's basically the polarity. So it becomes depolarized. So you can see here, you've got basically the inrush of that sodium, and now you're going all the way up to a plus 35. So that's a depolarization event. So your depolarization continues as that sodium begins to and continues to rush in, changing and flipping that charge. Repolarization basically happens right after depolarization. So this happens when the sodium channels close, disallowing sodium to completely rush, and the potassium channels open. So the potassium flows out of the cell, allowing you to rebalance the charge. And so ultimately what happens is the inside of the cell slowly becomes negative again as it resets. And the sodium potassium pump basically completes the action by essentially resetting and reestablishing the sodium gradient. So it loads the sodium back up there and resets the resting membrane potential. So the potassiums are basically returned to the inside of the cell where they were at the beginning. The sodium is going to be put right back to the outside of the cell where it was at the beginning. And so now you've got your resting membrane potential restored. So that's kind of what it looks like. So notice now you have all of your sodium channels are closed, right? So you have these guys all closed here, and your potassium channels are now open. So your potassium is able now to leak through and sort of bring the positive charge and sort of balance it out a little bit. And then ultimately what's going to happen is that's going to restore it along with your sodium potassium pump back to minus 70. Okay. So if we take a look at the graph of an actual neuron, right, the entire process of depolarization, repolarization, it's a rapid situation, right? So you're basically talking about a few milliseconds is how long it takes for this to occur. Now, to visualize that, we have to measure the voltage across a membrane, and we have to plot that over time. And this is kind of what it looks like. So bear with me as I double check on my systems to make sure that all systems are going, that I'm recording, and not just talking to myself. I apologize for that. So this is kind of what it looks like. So here's basically a measure of our voltage on the y-axis here. And then, of course, over time in milliseconds. And so you can see, you start down here at resting membrane potential, which is about minus 70 millivolts. And then as you increase, you notice you get up to threshold, which is basically this dotted line. And once you get to threshold, then you depolarize rapidly. As you get that inrush of sodium, that basically is your depolarization curve, the incline, the positive slope. So this is your sodium moves into the axon. Then what happens is you close all those sodium gates, your potassium opens up, you get repolarization or resetting of your system. You get a brief moment of what's called hyperpolarization, and then your sodium potassium pump brings it back down to resting membrane potential. This is what's happening every single time you receive an action potential. So let's go ahead and take a look at the propagation now. There's two different propagation styles. First of all, if you're an unmyelinated axon versus if you are a myelinated axon. If you're an unmyelinated axon, what happens is the action potentials will stimulate adjacent parts of the axon. And so this will basically produce an action potential in the adjacent membrane. So conduction here can be very slow, about 1 meter per second, because each section of axon has to be stimulated. So it moves very slowly. Now, propagation of an action potential can move through myelinated fibers much quicker because the action potentials don't go through each adjacent membrane. They actually jump from one node to another. This is what's called saltatory conduction. It's kind of like jumping or skipping. You can skip much faster than you can walk. And so this can move at about 100 meters per second. And so the myelination itself are self-propagating. So ultimately, once you get an action potential going, it generates another along the entire length of the axon, and it'll basically move to the other node. So this is kind of what it looks like. So here you can see an action potential occurring here, so that inrush of sodium. What it does basically is it moves underneath that myelin sheath and immediately jumps to the next node. So here you can see your next depolarization event. And then this is going to jump over the myelin sheath, and then it's going to depolarize the next node, and then it's going to jump over and then depolarize the next node. So this moves along much faster. This is saltatory conduction. This is how action potentials move in most of your peripheral nerves. Generally speaking, whenever you depolarize a piece of axon membrane, there's a period of inaction that's called the refractory period. So the refractory period is an area where you're unable to conduct another action potential. So you're insensitive to new stimulus. So what this does is it basically means you can't go backwards, right? So you can only go one direction, and that's forward, right? So then once you reach the axon terminus, you get to the axon terminal, right? That's basically where the axon is going to branch into. This area of fine branches called the axon terminals. Each terminals typically is going to be close to their target. Either it's going to be a dendrite of another neuron, or it's going to be the muscle cell itself. But typically at this juncture is what's called a synapse, which is a gap between the presynaptic and the postsynaptic cell. So the synapse, this gap is called the cleft, right? The synaptic cleft, and it separates the neuron from the receiving neuron. And in order to get across this cleft, you need a neurotransmitter, which is a chemical signal that basically transmits or carries the signal of these action potentials across the synapse, and then to the receiving neuron. And so they're harbored in that presynaptic cell in what's called synaptic vesicles. These are little vesicles that basically reside at the end of the axons, and they're full of neurotransmitter. This is what's happening at the axon terminals. So this is kind of what it looks like. So here's your sending neuron or your presynaptic cell. So typically speaking, what's going to happen is the action potential is going to ripple down the axon, and then it's going to hit these calcium channels, which are voltage sensitive. So when they see that membrane change, that potential change, they're going to open up and allow calcium to inrush. That calcium will then signal these synaptic vesicles to open up and to basically release their neurotransmitter in the synaptic cleft. That neurotransmitter will then bind to and open up new gates that are in the target cell, the receiving neuron, which will reignite that action potential in the target cell and allow it to basically communicate the message further. And so this is kind of what it looks like. So here you can see these little ligand-gated channels, which will allow then the inrush of sodium, and that will reignite or redepolarize that membrane. And then you're off to the races now in your target cell. So ultimately, at the synapse, right? So you have your nerve signals, which are going down the axon. And then that's going to allow these calcium ions to basically enter into the synaptic cleft. And excuse me, actually these ions to stimulate the synaptic vesicles to release their neurotransmitter, which is released by these synaptic vesicles into the cleft. And then they will bind to receptor proteins in the receiving neuron to reignite that action potential in the receiving one. And so in some cases, these communications can be excitatory or they can be inhibitory. So for instance, it depends on the type of neurotransmitter that we're putting in there for the receiving neuron, right? So they can either be an excitation signal or an inhibition signal. So it's excitation if the neurotransmitter causes a sodium openings, right? This kind of reignites depolarization. It's an inhibition signal if the neurotransmitter causes a potassium to exit, right? So if it's potassium exiting, then that's going to be an inhibitory signal because it does this the opposite. This is kind of what it looks like. So here you can see a neuron. So here's the cell body. This is an actual electron micrograph. So here's your little axon terminals that are all touching on this cell body of this neuron. So you can see you have all these different little contact points to the cell body. Some of them are on the cell body, some of them are on the dendrites, but you have these contact areas onto the cell body. Some of them are excitatory. So the green ones, for instance, are excitatory synapses telling it to do something. And then some of them are inhibitory responses. And so what the cell body has to do is basically sort through all these incoming stimuli, and then it basically sends out a final net response based on all the inputs. So ultimately, how do you get the neurotransmitter out of that cleft, right? Because otherwise you're going to have just indeterminate neural excitation or inhibition. So once your neurotransmitter has created the response, it has to be removed from the synaptic cleft. Now, typically speaking, there's enzymes that are in the synaptic cleft that will essentially break down the neurotransmitter. For instance, in a muscle synapse, you have acetylcholine in that gap. So instead of just giving you a constant contraction signal, acetylcholinesterase, which is an enzyme that comes in and breaks down acetylcholine, will basically break down that acetylcholine, and it'll basically reabsorb that into the sending membrane so that you can build more. So this will build more acetylcholine. And so you kind of clear out that synaptic cleft and get that out of there. The synapse itself, basically, you have essentially a short existence of neurotransmitters. You don't need them in there very long, just enough to reignite that action potential, and you got to pull them out, right? So what this does, it creates a situation where you don't have continuous stimulation, so you don't have constant contraction, for instance, right? So if you had neurotransmitter hanging on in that cleft, basically you wouldn't be able to respond to a new incoming signal. So you would be like forever in that refractory period. So in terms of neurotransmitters, we have a lot of them, right? So there's more than 100 neurotransmitters. Acetylcholine we just looked at, that's the one that we use for our musculature. We have norepinephrine. Dopamine is a common one, serotonin, glutamate, GABA. These are all different types of either nerve to muscle or nerve to organ or nerve to gland types of synapses that we have between these neurotransmitters. For instance, acetylcholine and norepinephrine are active in both the central and the peripheral nervous system. And in the peripheral nervous system, they typically act at neuromuscular junction, so basically associated with your skeletal muscle contraction. In the peripheral nervous system, your acetylcholine will essentially excite your muscle cells, and they'll also inhibit your cardiac muscle cells because basically they're trying to get your skeletal muscle cells to contract. Norepinephrine will excite your smooth muscle cells. And so that's going to be an important part for like your organs and your blood vessels. But in the central nervous system, norepinephrine is an important type of a communicator, especially like when you're looking at dreaming or waking or mood. So a lot of your kind of arousal cycles are driven by norepinephrine. Serotonin is involved in thermal regulation and also sleeping and emotions and perception. So there's a lot of different sorts of things that serotonin will be involved in. And a lot of drugs will oftentimes act at the synapse location. Some will mimic neurotransmitters, others will interfere with neurotransmitters depending on what kind of an effect you're trying to get to. So synaptic integration is a situation where you have the summing up of multiple incoming either excitatory or inhibitory signals. And so ultimately what's going to happen is if a neuron receives enough excitation signals and it overwhelms the inhibition signals, then ultimately it will basically transmit a signal through its axon. If, however, it receives more inhibitory over excitatory signals, then ultimately the sum of these will prohibit the axon from reaching threshold, and so you won't get a signal going through your axon. So you can use inhibitory and you can use excitatory to regulate what kind of a message, if at all, you're sending through the axon. So it's kind of what it looks like. So this is your resting membrane potential here at -70. As you see the orange line, this is an inhibitory signal, which will basically depress your signal, so making it harder to actually get up to excitation. So you're actually going below resting membrane potential. An excitatory signal will actually move you toward threshold, so getting you ready for depolarization. And so that's basically how excitation and inhibition works. It either gets you further away or closer to your threshold so you can depolarize. Okay. So let's go ahead and take a look then at the anatomy of the central nervous system. We're going to start off with the spinal cord and the brain. So essentially these are the guys who are receiving sensory information from the rest of your body. The spinal cord and the brain are essentially protected, first of all, by the vertebra and the skull, respectively. The spinal cord and the brain are essentially wrapped up in a layer of connective tissue liners called the meninges, and you have three different meningeal liners actually in the central nervous system. Inflammation of any of these liners is what causes meningitis, oftentimes if it's infectious meningitis associated with bacterial or viral infections. You also have in your central nervous system its own fluid, so cerebral spinal fluid, or CSF, is basically going to be flowing between these meningeal layers, and it helps to cushion and protect and to feed, in some cases, the central nervous system. For instance, when you do a spinal tap or a lumbar puncture, basically you're sticking a needle into your spinal cord and you're drawing out cerebral spinal fluid. So you're basically collecting CSF. And so in your cerebral spinal fluid, in your central nervous system, your brain in particular, you've got major cavities or openings, just like when you had your heart ventricles. You have 4 ventricles in the brain and the central canal of the spinal cord. And these basically are full of cerebral spinal fluid. And they circulate the cerebral spinal fluid throughout your brain and your spinal cord. So they're interconnected with each other and they circulate the cerebral spinal fluid. Okay. So cerebral spinal fluid itself, CSF, basically is a fluid. And eventually it's going to drain away into the cardiovascular system so you can kind of refresh it a little bit. You can create blockages. That can be an issue, right? So for instance, if you have accumulation of cerebral spinal fluid because of a blockage, it can lead to what's called hydrocephalus, or basically that means waterhead. So hydro means water, cephalus refers to your head, so this means waterhead. So there's water on the brain. And so if it collects in an adult, basically the brain cannot enlarge because our skull bones are fused together. So it sends a push against the skull. And this can create brain damage and it can be fatal. So anytime you create abnormal pressures inside your cranial cavity, you can create a lot of issues. So one of the reasons why if you have a hematoma in your brain from a brain injury, a lot of times what they'll do is they'll cut a hole in your skull to relieve some of the pressure to keep you from dying. You could go into a coma, you can go into seizures, you can do all sorts of bad things. So it's kind of what the ventricles look like of the brain. You can see they look like ram's horns. And so you can see you have 2 lateral ventricles. So the lateral ventricles tend to be ventricles 1 and 2. Beneath them, in the T-like structure, you're going to see a 3rd ventricle, which is number 3. And then below that is number 4, which is sandwiched between your cerebellum and your brainstem. And then that leads down into the spinal cord. So you can see how all these ventricles in the spinal cord, the central canal of the spinal cord, are all interconnected with each other. So then let's take a look at gray and white matter. So we mentioned this a little bit before. And so we want to also kind of fill that out a little bit. So gray matter basically is essentially a region of your brain where you contain a lot of cell bodies that are short and non-myelinated in nature, creating their grayness. White matter typically is myelinated because myelin is white because of the lipids in the myelin sheath. And they tend to have clusters of myelinated areas, and that creates these whitish tracts. And so that's where white matter comes from. The spinal cord will basically extend from the base of the brain through the foramen magnum, which is the large hole in your skull, at the base of your skull, where your spinal cord will go through, and then it travels through the vertebral column down to your tail. And so the cross-section of the spinal cord shows a couple of different structures in there. First of all, you see the central canal, which is connected to the ventricles of the brain. You also see gray matter and white matter. Remember, clusters of cell bodies and clusters of axons, basically. The spinal nerves will basically project from the cord through small openings called the intervertebral foramina. So foramina means little hole. That's what that means. And in between each of the vertebrae are intervertebral discs that separate the vertebra and basically are shock absorbers. And so if your disc ruptures or if it herniates, then it can compress the spinal nerve, can pinch down on the spinal nerve. That can create a lot of pain and also loss of mobility is what it looks like. So here's our cross section. So the butterfly looking structure is the gray matter, right? That's all cell bodies in there. We're doing lots of processing. And it's surrounded by white matter. So those are lots of axons, lots of transmission and things of that nature. You can see how the spinal cord itself is basically embedded in that vertebral canal of the actual vertebra itself. And you can see the spinal nerves, which are basically sticking out of each vertebra as it kind of goes through your spinal column. Now, on its way out of the spinal cord itself, you have two connections that become the spinal cord. The first one is on the dorsal side, right? That's your back. And so you have a dorsal, what's called a dorsal root that comes out of the dorsal side of the spinal cord, and it basically starts to swell on what's called the dorsal root ganglion. A ganglion is also a collection of cell bodies. It's a type of gray matter, a specific type. So this is basically an area that does a lot of processing. And so it'll go from the dorsal root ganglion, and then it'll merge with the other root, which is the ventral root, which is coming from the ventral side. And when they merge together, then they become the spinal nerve. And so that's basically your spinal cord. So your gray matter is typically in the middle. It's centrally located. Your dorsal root is going to be carrying a lot of sensory information entering the gray matter. The ventral root of the spinal nerve contains all the motor fibers. So these are the outgoing messages. So the dorsal root is going to have all of the incoming messages. So if you look at it that way, what's going to happen is sensory information is going to be coming from your body through the spinal nerve into the dorsal region. Then it'll go up through the brain and then back down through the spinal cord. And then the response will exit out the ventral side and go out the spinal nerve to communicate with your body. So when they join, they basically form the spinal nerve, which is typically a mixed sensory motor nerve. And that's part of your peripheral nervous system. So here's the meningeal layering that we can see here. So here's your dorsal side, here's your ventral side. So you can see your central canal is that little hole right in the middle. And so you can see your gray matter and your white matter. This is your sensory dorsal root with your dorsal root ganglion. This is your motor ventral root. And then this is your spinal nerve. Now, if you take a look at the layerings, you can see the different layers of meninges. So you have an outer layer called the dura mater, a middle layer, which is typically called the arachnoid mater, and then another layer that's just resting right on top, which is typically called the pia mater. So you have 3 different layers of the meninges. And so that's kind of your meningeal line. That basically follows not just your brain, but it all goes down your spinal cord as well. So you have the same types of meningeal liners around your spinal cord as well. So white matter basically surrounds your gray matter. And typically the white matter is going to have ascending tracts, basically. This is carrying information to the brain. And then the descending tracts taking information from the brain going down. And so oftentimes these tracts will cross as they exit or enter the brain, so the left side or the right side of the brain, and then they'll communicate to the opposite side of the body, right? For instance, if it's communicating with the right side of the brain, it's going to be going to the left side of the body and so forth. And so ultimately, this is kind of a gateway often for pain signals. So for instance, motor signals from the brain are basically going to pass down through the spinal cord and it's going to go out to the muscles. So your brain notices the central processor that's making the decision is what to do about all this pain that you're feeling. When you have paralysis, oftentimes you have a loss of sensation and voluntary control, right? So for instance, if you're not able to actually send in these sensory information, then what's going to happen is you're going to have loss of control, and ultimately you're not going to have the ability to move, right? So you're literally not just going to have that sensory information coming in. Okay. So in paraplegia, this is typically if you have a thoracic injury, usually it's affecting your lower body and your legs, usually. If quadriplegia is usually in the neck region, this is when it's going all the way down to all 4 limbs, both your pectoral girdle and your hip girdle. We do, however, have what's called a reflex arc, right? So the normal trajectory of your stimulus is to be picked up by your sensory neurons, sent to your spinal cord. It's going to move up through the ascending tracts for your brain to interpret. A message will be sent in response to that down the descending tracts back to your spinal cord, and then back out through the ventral root in order to stimulate a response to the muscles. That's how it usually works. However, reflex arcs will essentially short circuit the normal pathway. So you don't go to the brain. What happens is the stimulus will be picked up by the sensory receptors, generates a signal that goes to the axons in the spinal cord. Now what happens is interneurons basically will integrate the incoming data and immediately send a signal to neurons, basically bypassing the brain. So the brain's not making this decision this time, it's the interneurons. So they go directly to the motor axons, which then tell the skeletal muscles to contract. This can also be a reflex associated with smooth muscle, organs, or glands. This is helpful for those situations when, for instance, you need to do something and you don't need your brain to think about it. There's some things you want to have on autopilot. For instance, your blood pressure regulation. When it falls, increasing it, things of that nature. Internal receptors in your carotid arteries and aorta will send signals up the ascending tract to the cardiovascular center in the brain. But motor signals basically passing down the descending tract to the spinal cord, out to the blood vessels. They'll constrict and they'll increase your blood pressure. So this basically is a giant reflex arc. So oftentimes this is automatic and spontaneous. Now, it doesn't necessarily mean that you cut your brain out of it completely. You tell your brain what just happened, right? So for instance, if I put my hand on a hot stove, I don't have to think about how hot it is. I immediately reflexively pull my hand off the stove. That's because the sensation came directly from my hand to my spinal cord. It was processed by an interneuron and immediately acted upon muscles to pull my hand away. Well, at the same time that it's sending out the communication to my muscles, there's also a communication going up the ascending tracks to my brain know, oh, by the way, I basically took matters in my own hands and I told the hand to take itself off of the stove. So I just figured I'd just CC you on that communication. That way the brain doesn't have to make the decision. It just basically is made a decision. So reflexes oftentimes are either automatic response in your body or the responses where you have to actually make some sort of movement or because it's like a life or death sort of a thing. Okay. So the brain then, the structure of the brain, we'll take a look at the brain from here on out. So we're going to be taking a look at the different regions. So we have the major regions of the brain are the cerebrum, we have the diencephalon, the cerebellum, and the brainstem. So when you take a look then at the cerebrum, sometimes we'll refer to this as the telencephalon. This is the largest part of the brain where we have the area that's kind of last in line to receive that sensory input. But this is also our integrator, right? This is basically the one in charge of pulling all the levers for motor responses. And this basically is a big coordinator and a communicator of your nervous system, coordinating activities across all other parts of your brain. So the cerebellum is an important area. Excuse me, the cerebrum is an important area. So it's kind of what it looks like. So your cerebrum is all the wrinkly part associated with what's called the cerebral cortex. In the middle of that region, you're going to notice you have your ventricles lined by your corpus callosum, and then you have different areas of your brain. So for instance, this here is your cerebrum, and then in the middle is basically going to be what's called your diencephalon. This houses areas of your brain called the thalamus, which also has the third ventricle, so this is associated with the third ventricle, and also the hypothalamus, which is associated with your endocrine system. We'll see that in a couple of chapters. And then below that, you're going to have your brain stem, which is this piece here. And you're going to have three pieces to that. So you're going to have your midbrain, and then you're going to have your pons, which is this big swollen area. And then you're going to have your medulla oblongata, which is this piece here. And then you go to your spinal cord. And your cerebellum is going to be this little kind of cauliflower-looking structure at the base of your skull. Notice also, you see vasculature. So these are the ventricles. This is basically the spaces in between the meninges that circulates CSF. So you can see that blue space is basically the cavities, the areas where the CSF is being circulated around the brain and bathing the brain. So this is basically what your human brain looks like. It's divided into right and left hemispheres, or right and left brain. In the middle is going to be that deep groove, which is the longitudinal fissure. So that's this guy right here. And typically this will divide you into right and left cerebral hemispheres. Communication between right and left is via the corpus callosum, which if you take a look here, is this large structure right here that kind of looks like a little ram's horn. So that's going to be your corpus callosum. That's basically connecting the integration communication between your right and left halves. And then what we have also in the brainology, if you will, is two structures. One called the gyri or the gyrus, which is essentially a thick fold that's basically separated by shallow grooves called sulci or a sulcus. So essentially the ridges are the gyri and the grooves are the sulcus. So the cerebrum basically is divided into different lobes that mirror your skull bones, right? First of all, you have your frontal lobe that is most anterior underneath your frontal bone, the parietal lobe, which is right underneath your parietal bone, the occipital lobe, which is right beneath your occipital bone, and of course your temporal lobe, which is right around your temporal bone for your ear. This is what it looks like, right? So for instance, you have your frontal lobe, which is this region here in the front. And then you have your parietal region, which is this colorful region right here. Your occipital region, which is behind your occipital bone. Here and here, region up and of the hours